We are Senior Design Group 22, and our project is called the In Vitro Model of Traumatic Brain Injuries.
Traumatic brain injury is a leading cause of death in the U.S. 3.1 million people in the U.S. are currently living with disabilities from TBIs. Annually, 1.5 million people sustain this injury and 50,000 people die from the damage.
The goal of this model was to be able to reduce the need for in vivo animal studies, while also creating a more detailed visualization of traumatic brain injuries in terms of the human brain. In vitro models are ethical, cheap, repeatable, and allow for real-time observation, while in vivo models are expensive and morally suspect.
The scaffolds are donut-shaped, porous, and made of silk and were provided to us by a lab at Tufts University. The purpose of the donut shape is to resemble the laminar layers of the cortex. As shown in the bottom image, the cells are seated in the porous part, resembling the gray matter, and the collagen-filled hole in the middle is where the axons grow through and connect, representing the axon-only white matter. Prior to seeding the cells, the scaffolds are autoclaved, coated in poly-D-lysine to make it easier for the cells to attach, and then incubated in neurobasal medium. The top two images show the scaffold in the testing device.
The cells from the day 18 embryonic rat cortices are separated through processes including trypsin and DNase. Collagen is added to both the dish and the scaffolds a day after cells are seeded to enable axon growth. In the bottom image, it can be seen that by day 4, axon growth between cells create a neuronal network similar to that in the brain.
This is an overhead view of the device Dana created. The arrow points to the donut-shaped scaffold in the petri dish. It is ready to be struck by the hammer in this position. The mass attached to the spring, shown by the right arrow farther back, is released and causes the hammer to strike the scaffold from the right side and create an impact. The calibrated load cell, which is circled, measures the impact force.
Here is a demo test showing the device in action with a spring tension distance of 5 centimeters using a silk scaffold in place. Dana designed and constructed the testing device to hold the scaffold for force testing.
After culturing the cells for a week, mechanical testing was performed on the silk scaffolds seated with the rat cortex. Images were taken during this process with a high-speed camera, which captures 800 frames per second. These 9 images show the process of the microhammer striking the scaffold during one trial. The main part of this collision, which is shown on this slide, only took 9 frames or 10.8 milliseconds. The device outputs force data, which is calibrated to newtons. We then graph our results of force versus time. 356 data points are taken in 63 milliseconds. Both force graphs have similar trends and experience definite peaks around the same time. This means that our device is functional and provides repeatable results.
After testing, the scaffold is fixed with 4% formaldehyde and incubated overnight. The next day, the scaffold is treated with a buffer to permeabilize the cells, and anti-beta tubulin primary antibody shown on the left is added. The next day, cells are treated with anti-fibronectin secondary antibody shown on the right to allow for fluorescence when visualized. On day 4, the cells are washed with PBS and imaged using the confocal microscope.
Confocal microscopy allows for greater magnification, fluorescence imaging, and viewing of the 3D layers of a sample. We use the confocal images to observe the damage to the neural network after impact. The video on the left shows the neural network that was formed 5 days after initial cell seeding. It is fully intact as it had no force applied to it. The cell bodies came together and formed a spheroid, while the visible axons formed an interconnected neural network. The video on the right shows a scaffold after force was applied to it. There are many pieces of axons seen throughout the scaffold, showing that there was axon breakage and the interconnected neural network was damaged.
We have created an optimal testing process that applies adequate damage to the seeded scaffolds. Axon growth and neural network formation was successful, as shown in the confocal image. The standing protocol allowed for visible neural network damage after impact. The high-speed imaging process was also successful and was used for strain mapping. The scaffold experienced the highest strain on the side that it was impacted. The device has also been calibrated and tested for feasibility. It is now ready to collect more data.
In the future, more trials will be conducted. By replicating the experiment, our team would receive more data, allowing us to be more confident in our results. We would also like to increase the number of hits per trial. Traumatic brain injuries can occur as a result of multiple hits to the head, and by testing multiple hits on the scaffolds, we can analyze how the different number of hits, possibly at different forces, affect the cells. Our results give us a better understanding of traumatic brain injuries in humans, which will lead to drug development and reducing animal testing significantly.
We'd like to thank David Kaplan, Stern Family Professor of Engineering at Tufts University, Dr. Mindy Tang Schomer of Jackson Laboratory, our immediate advisor, Professor Hoshino, and lastly, we'd like to thank the previous Senior Design Group members of 2018.